I've got a mug of peppermint tea, and I want to talk about celebrating the little wins. Grad school and science in general can be full of little and big failures. Sometimes you run an experiment and you do the very best job that you can and it doesn't work and it fails. Or sometimes you run an experiment and you mess up something stupid and it doesn't work and it fails. Or sometimes something crazy happens and science fails. And these can be little things or big things, but they pile up after a while and it can get to you. I have a feeling that there are going to be a lot of tea break videos inspired by those failures and those moments that kind of suck. So I want to talk instead about the opposite, about the times when good things happen. I've been trying to do a really good job this year of appreciating the little wins. Anytime something goes right for me in science, I'm trying to count it as a win. This can be something really small. This can be coming into lab and realizing that my cells aren't dead. That is a win. It doesn't have to be something as big as a full experiment working or finding a great result. I think I'm just trying to do a good job of being very mindful of the times when stuff works and appreciating those times and letting myself feel confident and happy about them, because I might normally sort of punch those feelings down. But I was inspired to talk about this because of a little win that I had in lab last week that I tried to record here. So it is 10:30 on a Wednesday night, and I am in lab, and I have been here since 8:45 this morning because I had a ton of stuff that I wanted to get done, but I'm totally okay with that because I've been working on an experiment for about a week or so, and it's not a big experiment, it's really a small thing. I was doing some sequencing, but I came up with sort of the sequencing scheme on my own, and I just sort of went forwards and I did it. I amplified the regions I wanted to sequence, and then I sequenced them myself, and then I did all of the computational stuff to take the sequencing and turn it into something that I could visualize on my computer and align the reads to the genome. And this is not hard stuff at the moment in the state of genomics, but it's not stuff that I do all the time. I am definitely a wet lab scientist primarily, and so the dry lab stuff, the computational stuff, is sometimes a little scary to me. And so I just got back preliminary results, and I think it worked. I think I did it right. I think I designed the right thing. I think I got the output that I wanted. It's still running somewhat, so, I'll figure most of that out later. But it's good. It's really good. I think I need to revel in the fact that I'm a little proud of myself right now, because science is so hard and science beats you down all the time because experiments just fail. You can design a great experiment and they fail for all these different reasons. There are so many things that are out of your control that, sometimes I think even if I have just a little win, it's just a little win. This isn't a big thing. I didn't discover something new. I just sequenced a thing the way I wanted to sequence it. But I have to really revel in the little wins. It's all the little wins that I think keep you going. Now, the kind of sad thing is that I came into lab the next day, and when I actually analyzed the data, it didn't work as well as I thought it had. It worked though, and so I was still happy. I was still happy that I brought it from sort of conception of experiment to the end of experiment, and I learned something from it, and that's the most important part, is that I got to the end, I learned something, and now I have tweaked that something, and I'm going back and I'm redoing the experiment with the better parameters, trying to learn something new. So it was all good. It was all good in the end. But I was just trying so hard in that moment to savor the fact that I had done something and it had worked, and it was a little win, even if it was just a little win. I think that it's important to try and, really hold on to those. Maybe this sounds a little silly, or maybe you're in a situation in science or in life where you get what I'm saying and you try and hold on to those little wins too, because they add up. And you can't just wait to be happy for the big wins, when you find something that's going to win you the Nobel Prize. If you wait for that, you're going to be real unhappy in science. You have to take happiness and pleasure in actually doing the science too, and I think a lot of that comes from appreciating when things do work and appreciating when you've done a good job. And it's not bragging, because I sometimes feel like I don't want to feel this because, it's bragging, I did a good thing. No, it's not bragging. I think it's just being personally happy about having done a good experiment. Trying to take pride in that. Let me know in the comments if you have had your own little win this week or big win. you know, in lab or in life, either one. I really hope that tea break can be a way for us to sort of come together as a community and talk about these things. If you're a scientist or if you're not a scientist, I think that a lot of people share these same things, and so I'm looking at it certainly through the lens of grad school. And so if there are other grad students, please come talk to me in the comments about your grad school experiences so that we're not all alone. I don't know why I'm saying that. And then remember to go forth and do science.